Hello everyone, and welcome to today's presentation. I'm a SOC, Prof. Yasser Madany, and I'm excited to share this session with you. I'm a senior member of IEEE and a senior member of URSI. Today, we'll be exploring a fascinating topic related to our field. Over the next few moments, I'll walk you through the key concepts, insights, and advancements that are shaping our industry. So let's get started and dive into the details together. Let's take a closer look at the overview of brain signaling. At its core, brain signaling forms the foundation of neural communication, which is essential for how the brain processes information and controls our behavior. It all starts with the basic building blocks, neurons and glial cells, working together to send and receive signals. These signals happen in two main ways: electrical signaling, where action potentials generate rapid impulses, and chemical signaling, where neurotransmitters cross synapses to pass the message along. These signals are then integrated and processed across complex networks within the brain. Different regions like the cortex, hippocampus, and cerebellum each have specialized roles, helping us perceive the world, remember information, and coordinate movements. The brain's plasticity and capacity to learn come from its ability to strengthen or weaken these connections over time, allowing us to adapt. However, when signaling goes awry, it can lead to disorders such as epilepsy, Parkinson's disease, or depression. Advances in technology, especially brain-computer interfaces, are now tapping into these principles to create direct communication links between neural activity and external devices. This is opening exciting new possibilities in medicine and how we interact with technology. Let's begin with an overview of how the brain communicates, which is fundamental to understanding its incredible capabilities. The building blocks of brain communication include electrical and chemical signaling, both of which work together to transmit information quickly and efficiently. Electrical signaling involves impulses traveling along neurons, while chemical signaling uses neurotransmitters to pass messages across synapses. These signals aren't just simple on-off switches. They are integrated and processed by various brain regions, each with specialized roles that contribute to our thoughts, emotions, and actions. A key aspect of this communication system is plasticity, the brain's ability to adapt and change through learning and experience. However, when signaling pathways malfunction, it can lead to disorders such as epilepsy, depression, or neurodegenerative diseases. Understanding these mechanisms also opens the door to innovative technologies like brain-computer interfaces, or BCIs, which connect the brain directly to external devices. All of these points tie into our broader theme of exploring how the brain's communication networks underpin everything we do. Now, with this foundation in mind, let's move forward and delve deeper into each of these fascinating topics. Welcome to our next topic: Introduction to Brain Signaling. Today, I want to take you through how our brain communicates internally. This process is fundamental to understanding how we think, feel, and act. At its core, brain signaling involves a complex network of neurons working together seamlessly. Neurons are the primary cells responsible for transmitting information throughout the brain and nervous system. They communicate through electrical and chemical signals, which is what we call neural signaling. This signaling process allows us to respond to stimuli, process information, and coordinate actions. Understanding these signals is crucial because they underpin everything from basic reflexes to complex decision-making. Throughout this presentation, we'll see how these signals form the foundation for all brain functions and behaviors. So, let's dive deeper into how this intricate communication system operates and why it's so vital. This slide doesn't contain enough information. That's why we're moving to the next slide. The human brain is truly a remarkable and complex organ. It's made up of approximately 86 to 100 billion neurons, which is an astonishing number. These neurons are the fundamental units that enable our brain to function. They communicate with each other through electrical and chemical signals, which is how all the magic happens. These signals are what allow us to perceive the world around us, think through problems, move our bodies, and even experience emotions and feelings. This process of signaling is at the heart of brain function and is essential for everything we do. As we move forward in this presentation, we'll dive deeper into how these signals are generated and transmitted. Understanding brain signaling is key to grasping how the entire nervous system operates and how our brain controls our body and mind. It's the foundation of everything we'll explore in the chapters to come. Neurons, the building blocks of brain communication, are truly fascinating. They are specialized cells designed to transmit information quickly and efficiently throughout our nervous system. Think of them as the messengers that carry signals from one part of the brain to another, or from the brain to the rest of the body. Each neuron has a unique structure, with a cell body, dendrites, and an axon that work together to send and receive electrical signals. What's really remarkable is how neurons communicate through these electrical impulses and chemical signals. This process underpins every thought, feeling, and action we have. Without neurons functioning properly, our ability to think, learn, and even move would be compromised. This slide highlights how central neurons are to brain function, tying into the overall theme of understanding how our brains process and transmit information. Recognizing their role helps us appreciate the incredible complexity and efficiency of our neural networks. Let's take a closer look at the structure of a neuron. First, we have the dendrites, which are like the neuron's antennas, receiving incoming signals from other neurons or sensory cells. These signals then travel to the cell body, also known as the soma, where the neuron processes this information. Think of the soma as the control center, deciding what to do with the incoming data. From there, the signal moves along the axon, which acts like a long transmission cable, carrying the message to other neurons or target cells. At the end of the axon, we find the axon terminals. These are crucial because they release neurotransmitters into the synapse, the tiny gap between neurons, enabling communication across the neural network. Now, looking at the different types of neurons, we have sensory neurons that carry signals from our sensory organs, like our eyes, ears, and skin, up to the brain. Then there are motor neurons, which take commands from the brain and spinal cord to our muscles, enabling movement. Lastly, interneurons connect neurons within the brain and spinal cord, playing a vital role in processing and integrating information. All of these neurons are the building blocks of how our brain communicates, allowing us to perceive, think, and act. They work together seamlessly to keep our nervous system functioning smoothly. Let's take a closer look at the main parts of a neuron. First, we have the cell body, also known as the soma. This is the central part of the neuron that contains the nucleus and other organelles. It's responsible for keeping the cell alive and functioning, as well as processing the information it receives. Next, we have the dendrites. These are branch-like extensions that reach out from the cell body. Their main job is to receive signals from other neurons and then transmit those signals toward the cell body for processing. Think of them as the neuron's antennae, gathering information from the surrounding environment. Moving on, we have the axon. This is a long, thin projection that carries electrical impulses away from the cell body. Some axons can be quite long, connecting different parts of the nervous system that are far apart, which is essential for rapid communication across the body. Finally, there's the myelin sheath. This is a fatty layer that covers some axons, although not all neurons have it. Its key role is to increase the speed at which signals travel along the axon, making communication much more efficient. Understanding these parts helps us see how neurons function as the fundamental units of our nervous system, transmitting information quickly and precisely across the entire body. Now let's take a closer look at some of the main parts of a neuron. First, we have the nodes of Ranvier. These are small gaps in the myelin sheath along the axon. Their role is pretty important because they help speed up the transmission of nerve impulses. By jumping between these gaps, the signals can travel much faster down the neuron. Next, we have the axon terminals, also known as synaptic boutons. These are located at the ends of the axon. Their primary job is to release neurotransmitters, which are chemical messengers that communicate with other neurons or muscles. This is how signals are passed along from one cell to another. Finally, there's the synapse, which is the junction or connection point between two neurons. Specifically, it's where the axon terminal of one neuron meets the dendrite of another. This is the critical site where neurotransmitters are released to pass the signal from one neuron to the next, ensuring the flow of information throughout the nervous system. All these parts work together seamlessly to facilitate rapid and precise communication within our nervous system. This slide doesn't contain enough information. That's why we're moving to the next slide. Now let's take a closer look at how signals are transmitted in neurons, step by step. First, during the resting potential, the neuron is essentially at rest, with a negative charge inside the cell membrane compared to the outside. This creates a kind of electrical balance that's ready to be disturbed when a signal comes in. Next, the neuron receives a chemical signal, called a neurotransmitter, from another neuron at the dendrites. Think of dendrites as the neuron's antennae, picking up messages from neighboring cells. If this chemical signal is strong enough, it triggers an electrical impulse known as an action potential at the axon hillock, which is the base of the axon. This is the neuron's way of converting a chemical message into an electrical one to keep the signal moving. Once initiated, the action potential travels down the axon as a wave of electrical change. It's like a domino effect, where each part of the axon quickly depolarizes and repolarizes in sequence. In neurons that are myelinated, this process gets even faster. The electrical signal jumps between gaps in the myelin sheath called the nodes of Ranvier. This jumping, known as saltatory conduction, significantly speeds up the transmission of the signal along the neuron. All of these steps work together to ensure that information moves efficiently from one neuron to another, ultimately allowing the nervous system to coordinate complex responses and behaviors. Let's take a closer look at sodium ions. Sodium ions are essential players in our body's fluid balance and nerve function. They carry a positive charge, which makes them crucial in transmitting electrical signals across nerve cells. This process is vital for muscle contractions and communication within our nervous system. Understanding how sodium ions move and are regulated helps us appreciate their role in maintaining overall homeostasis. Throughout this presentation, we see how these ions are interconnected with other processes, highlighting their importance in health and disease. Now let's take a closer look at how signals are transmitted in neurons, step by step. When an action potential travels along the neuron and reaches the axon terminals, it triggers a very important process. This causes vesicles filled with neurotransmitters to fuse with the cell membrane. Essentially, these vesicles release their chemical contents into the synaptic cleft, which is the tiny gap between neurons. This release of neurotransmitters is crucial because it allows the signal to continue its journey. The neurotransmitters then bind to specific receptors on the dendrites of the next neuron. This binding starts the process all over again, propagating the signal. Understanding this step-by-step process helps us see how information flows efficiently through our nervous system, connecting individual neurons into a complex communication network. Let's take a closer look at the structure of the cell body, or soma. The outer boundary of the soma is called the plasma membrane, and it acts like a protective barrier. It encloses the cytoplasm, which is a gel-like substance filled with various organelles that carry out essential functions for the cell's survival. Moving inward, we find the nucleus, which is quite large and centrally located within the cell. This is where the cell's genetic material, DNA, is stored. The nucleus is surrounded by a nuclear envelope, which helps protect and regulate what enters and exits. Inside the nucleus, there's a nucleolus, a small but important structure responsible for producing ribosomes. And speaking of the cytoplasm, it's not just a filler. It contains numerous organelles that perform specialized tasks, all working together to keep the cell functioning properly. Understanding these key structures helps us grasp how cells operate as the fundamental units of life. Let's take a closer look at the cell body, or soma, structure of a neuron. One of the most critical aspects here involves the organelles that keep the neuron functioning properly. First, we have the mitochondria, which are often called the powerhouses of the cell. Their main role is to produce energy in the form of ATP, which is essential for all neuron activities. Without sufficient energy, neurons can't do their job effectively. Next, the endoplasmic reticulum, or ER, comes in two forms. The rough ER, which is studded with ribosomes, these are like tiny factories that produce proteins. This is especially important because neurons need a lot of specific proteins for maintenance and signaling. The smooth ER, on the other hand, is involved in lipid synthesis and detoxification processes, helping to keep the cell healthy and adaptable. Then there's the Golgi apparatus. Think of it as the cell's packaging center. It processes and packages proteins and other molecules, preparing them for transport or secretion. Ribosomes are also present. They're the actual sites of protein synthesis, building the proteins that the neuron needs to function properly. Lysosomes play a cleanup role. They digest waste materials and keep the cell free of debris, which is vital for maintaining cellular health. And let's not forget the Nissl bodies, which are clusters of rough ER and ribosomes. These are unique to neurons and are particularly important for protein synthesis, especially since neurons often need to produce large amounts of proteins to support their complex functions. Overall, these organelles work together seamlessly to ensure the neuron is energized, well-maintained, and capable of transmitting signals efficiently, key aspects in understanding how neurons support our entire nervous system. Now, let's take a closer look at the structure of the cell body, also known as the soma. One key component here is the neurofibrils. These are essentially a network of fine threads, part of the cytoskeleton, that serve two main purposes. First, they provide structural support to keep the neuron stable and maintain its shape. Second, they play a crucial role in transporting substances within the neuron, ensuring everything moves efficiently from one part to another. Moving on, the cell body also contains other inclusions. For example, pigment granules like lipofuscin, which are essentially waste products that accumulate over time. These can give us clues about the age and health of the neuron. Additionally, there are glycogen granules, which are important for energy storage. Think of them as the neuron's energy reserves, ready to be used when needed to support various functions. Connecting this all back to our overall theme, understanding these structural components helps us appreciate how neurons maintain their integrity and function, which is vital for nerve communication and overall nervous system health. Let's take a closer look at some of the key functions of neurons as outlined in this table. First, metabolism is fundamental because it produces energy and keeps the cell healthy, essentially fueling everything else the neuron needs to do. Without proper metabolism, the neuron can't function effectively. Next, protein synthesis is crucial for creating proteins that are involved in neurotransmission and repair. These proteins are like the workhorses that enable communication between neurons and help repair any damage that might occur over time. Then we have gene regulation, which controls cell activity through DNA and RNA. This process ensures that the neuron responds appropriately to various signals and maintains its proper function. Signal integration is another vital function. It's how the neuron combines inputs from dendrites to decide whether to fire an electrical signal. Think of it as the neuron's decision-making process, integrating all incoming information to determine the next action. Structural support is about maintaining and nourishing dendrites and axons, which are essential for receiving and sending signals. Without this support, the entire communication network within the brain would break down. Waste management is also key. Breaking down and recycling cellular waste keeps the neuron clean and prevents buildup that could impair its function. All of these processes are interconnected, and they work together within the framework of DNA, the blueprint for the neuron's entire structure and function. Understanding these functions helps us appreciate how complex and efficient our neural systems truly are. Let's talk about electrical signaling, and specifically about action potentials. These are crucial for how neurons communicate. An action potential is essentially an electrical impulse that travels along the nerve cell. It starts when a neuron is stimulated beyond a certain threshold, causing a rapid change in the electrical charge across its membrane. This change is what we call depolarization. Then, shortly after, the membrane repolarizes as potassium ions exit the cell, restoring the resting state. This entire process is quick and self-propagating, meaning once it starts, it moves along the neuron without losing strength. Understanding action potentials is key because they form the foundation of neural communication, allowing signals to travel across long distances within the nervous system. This is a perfect example of how electrical signals facilitate complex functions like sensation, movement, and cognition. Let's talk about the resting potential first. Neurons maintain a voltage of about -70 millivolts across their membrane when they are at rest. This is like a baseline electrical charge that keeps the neuron ready for action. Now, when a neuron receives a stimulus, it can trigger an action potential. This happens when the neuron depolarizes, meaning the inside becomes less negative. During this process, sodium channels open, allowing Na+ ions to rush into the cell. This influx makes the membrane potential positive, essentially flipping the electrical charge. Following this, repolarization occurs. Potassium channels open, and K+ ions exit the neuron. This movement restores the negative charge inside the cell, bringing it back to its resting state. After firing, the neuron enters a refractory period, during which it resets itself, ensuring it's ready to fire again when needed. Think of this wave of electrical activity traveling down the axon much like a row of dominoes falling. One triggers the next in a chain reaction. It's also important to remember the all-or-none principle. A neuron either fires completely or not at all. There's no in-between. Once the threshold is reached, the full action potential occurs. This entire electrical impulse moves down the axon like a wave, carrying information rapidly through the nervous system. In summary, these electrical signals, action potentials, are fundamental for communication within the brain and body, allowing us to perceive, think, and respond. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at chemical signaling, specifically synaptic transmission. This process is fundamental to how neurons communicate with each other. When an electrical signal, or action potential, reaches the end of a neuron, it triggers the release of chemical messengers called neurotransmitters. These neurotransmitters then cross the synaptic gap, the tiny space between neurons, and bind to receptors on the neighboring neuron. This binding can either excite or inhibit the next neuron, depending on the type of neurotransmitter involved. This process is incredibly precise and efficient, allowing us to perform complex functions like thinking, feeling, and moving. It also highlights the importance of chemical signaling in maintaining the overall communication network of the nervous system. Understanding synaptic transmission is key to grasping how signals are propagated and modulated throughout our bodies, which ties back to our broader discussion of neural communication and brain function. Let's take a closer look at synapses, which are the gaps between two neurons. This is where the magic of communication happens. When a neuron wants to send a message, it releases special chemicals called neurotransmitters from its presynaptic end. These neurotransmitters then travel across the synapse and bind to specific receptors on the next neuron, known as the postsynaptic neuron. Some common examples of neurotransmitters include dopamine, serotonin, acetylcholine, and glutamate. Each of these plays a unique role in how signals are transmitted and processed in our nervous system. Now, these signals can be either excitatory or inhibitory. Excitatory signals, like those from glutamate, increase the chances that the next neuron will fire, essentially encouraging the message to be passed along. On the other hand, inhibitory signals, such as GABA, decrease the likelihood of firing, helping to regulate and balance neural activity. This chemical signaling process, called synaptic transmission, is fundamental to how our brain communicates and functions. It's the core mechanism behind everything from muscle movement to complex thought processes, tying into the overall theme of understanding how our nervous system works at a cellular level. Now, let's take a closer look at signal integration and processing. This is a critical step in understanding how our system interprets various inputs. Essentially, it involves collecting signals from different sources, which could be sensors or other data streams. The key here is how these signals are combined and analyzed to form a coherent picture. Processing these signals thoroughly allows us to filter out noise, emphasize relevant data, and ultimately make accurate decisions. Think of it as the brain's way of synthesizing information from multiple senses to understand what's happening around us. This integration is vital because it ensures that the system responds appropriately, based on the most accurate and comprehensive information available. It connects directly to our overall goal of creating intelligent, adaptive systems that can operate efficiently in complex environments. So, by mastering signal integration and processing, we're enabling our systems to be more precise, reliable, and responsive, key factors in advancing our overall technology. Neurons receive multiple signals from other neurons, and these signals can combine in different ways to influence whether the neuron will fire an action potential. One important concept is temporal summation, which happens when signals arrive close together in time, effectively adding up to reach the threshold. On the other hand, spatial summation involves signals coming from different locations on the neuron, also combining to determine if the threshold is crossed. If the total combined signal exceeds that threshold, the neuron fires an action potential, sending a message down its axon. Now, moving beyond individual neurons, we see how neural networks come into play. Groups of neurons form circuits that work together to process complex information. These circuits are organized into specialized regions, like the visual cortex for processing visual information or the auditory cortex for sound. These regions are crucial because they enable the brain to handle different types of sensory input efficiently. All of this ties back to the overall theme of how the brain integrates and processes information. By understanding how signals are summed and how neural networks are organized, we gain insight into the incredible complexity and efficiency of our nervous system. Let's take a closer look at brain regions and their roles. As we explore this, it's important to understand that different parts of the brain have specialized functions, and they work together to shape our behavior and cognition. The brain is like a highly organized network, with each region contributing to our overall mental processes. For example, the prefrontal cortex is crucial for decision-making and impulse control, helping us plan and think ahead. The amygdala, on the other hand, plays a key role in processing emotions, especially fear and pleasure. The hippocampus is essential for memory formation, allowing us to learn from our experiences. By understanding these core areas, we can see how complex and interconnected brain functions are, and how disruptions in specific regions can influence our mental health. This knowledge ties back to the overall theme of understanding the biological basis of behavior and cognition. Signals travel through white matter tracks, which act like the brain's communication highways, connecting different regions so they can work together seamlessly. Each part of the brain processes specific types of information. For example, the occipital lobe primarily handles visual signals, helping us interpret what we see. The temporal lobe is responsible for auditory information and memory, so it plays a key role in hearing and remembering. The parietal lobe processes sensory inputs and spatial information, which helps us understand our surroundings and body position. Meanwhile, the frontal lobe is crucial for decision-making and motor control, enabling us to plan actions and move intentionally. Different brain regions have specialized roles that support our overall functioning. The cerebral cortex, for instance, is involved in higher-order functions like thinking and planning. The thalamus acts as a relay station, routing sensory information to the appropriate areas for processing. The hippocampus is essential for forming new memories, while the amygdala helps us process emotions. The cerebellum coordinates movement and maintains balance, ensuring our actions are smooth and coordinated. By understanding these different regions and their roles, we can see how the brain integrates various types of information to produce coherent thoughts, feelings, and actions, highlighting the incredible complexity and specialization within our brain's network. Let's take a closer look at brain regions and their roles. As we explore this slide, I want to emphasize how different parts of the brain work together to influence our behavior and cognition. The brain isn't just one uniform organ. It's a complex network where each region has a specific function. For example, the prefrontal cortex is crucial for decision-making and impulse control, helping us plan and regulate our actions. The amygdala, on the other hand, is heavily involved in processing emotions like fear and pleasure, which can drive our responses to various stimuli. Moving to the hippocampus, it plays a key role in memory formation and spatial navigation, allowing us to learn from experiences and find our way around. Understanding these regions is essential because it helps us see how different parts of the brain contribute to the overall picture of human behavior. This interconnectedness is a core theme of our presentation, recognizing how the brain's structure underpins function and ultimately how it shapes who we are. Let's take a moment to understand how brain memory actually works. When we think about memory, it's not just about storing information like a hard drive. Instead, it involves a complex process where different parts of the brain communicate and work together. First, we have encoding, which is how we initially take in information. Then, there's storage, where that information is maintained over time. And finally, retrieval, which is how we access that information when we need it. One key point to remember is that these processes are interconnected and rely on neural networks. When we learn something new, synapses in the brain strengthen, creating pathways that make future recall easier. This process is fundamental to understanding how learning and memory are so dynamic. Connecting this back to the overall theme, understanding these mechanisms highlights the importance of how we engage with information, whether through practice, repetition, or meaningful connections, to reinforce our memory. It's a reminder that building strong memories is an active process, not just passive storage. Let's take a closer look at brain memory types. When we talk about memory, we're really referring to the different ways our brain stores and retrieves information. There are several key types, each serving a different purpose. First, we have sensory memory, which is very brief and captures information from our senses right after an event occurs. Think of it as a quick snapshot. Next is short-term memory, also called working memory. This is where we hold information for a few seconds to minutes, enough to work with it or make decisions. Then, we have long-term memory, which stores information over extended periods, from hours to a lifetime. Long-term memory includes both explicit or conscious memories like facts and events, and implicit memories, which are unconscious skills like riding a bike. These different memory types work together to help us learn, adapt, and function daily. Understanding these distinctions is crucial because it highlights how our brain processes and preserves information, tying into the overall theme of how memory shapes our experiences and learning processes. Neural oscillations, also known as brain waves, are rhythmic or repetitive patterns of neural activity that occur in the central nervous system. These waves are generated by the synchronized activity of neurons working together. If you look at EEG recordings, you can actually observe these oscillations directly. The key to understanding where these oscillations come from lies in the interactions within neural networks. They arise from excitatory and inhibitory feedback loops between neurons, which help regulate the timing and coordination of neural firing. Additionally, paced firing of neurons within specific networks, like the thalamocortical circuits, plays a crucial role in producing these rhythmic patterns. Ion channel dynamics and changes in membrane potentials are also fundamental, as they influence how neurons fire rhythmically. These oscillations aren't confined to just one spot. They can occur locally within a specific brain region or globally, spanning across multiple regions of the brain, coordinating activity on a larger scale. So, in essence, neural oscillations are a fundamental aspect of brain function. They help synchronize activity across different parts of the brain, which is essential for processes like perception, attention, and even consciousness. They truly highlight the rhythmic harmony underlying our neural activity. Let's take a closer look at how different brain waves relate to our consciousness and focus. When we're actively engaged in problem-solving or concentrating, our brain produces specific wave patterns. For example, during moments when we close our eyes to relax or meditate, certain waves become more prominent. These states are closely tied to memory encoding, helping us process and store information effectively. Understanding these different types of brain waves gives us valuable insight into how our brain functions across various mental states, which ties back to the overall theme of how our brain's electrical activity influences our cognition and awareness. Neural oscillations are often thought of as just background noise, but in reality, they play some really critical roles in how our brains function. For example, in communication, these oscillations help synchronize activity between different brain regions, like the cortex and hippocampus, which is essential for coordinated processing. When it comes to attention, alpha waves are particularly interesting. They act like a filter suppressing irrelevant stimuli so we can focus on what truly matters. Memory is another area where neural oscillations are vital. The coupling between theta and gamma waves is crucial for encoding new information and retrieving stored memories. Moving to motor control, beta rhythms help regulate voluntary movements, ensuring smooth and controlled actions. Lastly, in terms of consciousness, gamma oscillations are closely linked to awareness and the integration, or binding, of sensory information, which helps us have a coherent experience of the world around us. All of these points highlight how neural oscillations are not just passive rhythms. They are actively shaping how we communicate, focus, remember, move, and experience consciousness. They truly serve as the brain's dynamic toolkit for supporting complex functions that define our cognition and behavior. Now, let's talk about how different oscillations in the brain interact through a process called cross-frequency coupling. One key mechanism is phase-amplitude coupling, or PAC. Here, the amplitude of a fast wave, like gamma, is modulated by the phase of a slower wave, such as theta. Basically, the slow wave's phase influences how strong the faster wave's activity is at any given moment. Another important mechanism is phase-phase coupling, where two waves synchronize their phases. When this happens, their peaks and troughs line up, creating a coordinated rhythm across different frequencies. Then there's amplitude-amplitude coupling, where two waves influence each other's strength, fluctuating together over time. All of these processes are crucial because they allow the brain to coordinate activity across multiple scales. This multi-scale coordination is fundamental for complex functions like perception, memory, and decision-making, tying back to our overall theme of understanding how the brain integrates information across different levels. Let's take a closer look at how neural oscillations are studied. We have several key methods to consider. First, EEG, or electroencephalography, which is a non-invasive technique that records electrical activity directly from the scalp. It's widely used because it's accessible and relatively easy to implement. Similarly, MEG, or magnetoencephalography, measures magnetic fields produced by neural activity, also non-invasive and providing excellent temporal resolution. These methods are essential for capturing neural oscillations in real time. Now, for more invasive recordings, we have local field potentials, or LFPs. These are typically obtained in animal studies or during surgical procedures, where electrodes are placed directly into brain tissue. LFPs give us detailed information about local neural activity, but they are more invasive. When analyzing the data collected from these recordings, spectral analysis plays a crucial role. Techniques like Fourier transforms or wavelet transforms help us understand the frequency content of the neural signals. This is important because different oscillation frequencies are associated with various cognitive and behavioral states. Overall, these tools and techniques allow us to measure and analyze neural oscillations effectively, helping us uncover the underlying dynamics of brain activity that are so vital to understanding cognition and consciousness. let's explore the concept of plasticity and learning. Plasticity refers to the brain's incredible ability to adapt and change in response to experience. It's a fundamental aspect of how we learn new skills or recover from injuries. When we talk about learning, we're really talking about how neural connections strengthen or weaken over time through this plasticity. The more we practice a skill, the more those neural pathways become reinforced, making the behavior more automatic. This dynamic ability to reorganize itself highlights how adaptable our brains are throughout life, not just in childhood. Connecting this to the broader theme of the presentation, understanding plasticity helps us appreciate how learning isn't just about acquiring new information. It's about physical changes in the brain that support those changes. So, in essence, plasticity is the foundation that enables us to continuously grow and adapt, making learning a lifelong process. Neuroplasticity refers to the brain's incredible ability to reorganize itself throughout our lives. It's what allows us to adapt, learn new skills, and recover from injuries. When we talk about synaptic plasticity, we're referring to how the connections between neurons can strengthen or weaken over time based on how frequently they are used. This is often summarized by Hebbian theory, which states that cells that fire together, wire together. So, the more we activate a particular pathway, the stronger those synaptic connections become. On the other hand, structural plasticity involves the growth of new connections and even new neurons, which adds to the brain's capacity to change and adapt. All of this ties into how we learn, form memories, and recover from brain injuries. It's a dynamic process that highlights just how adaptable our brains are, emphasizing the importance of experience and practice in shaping our neural networks. Connectomics is the scientific field that focuses on mapping out how neurons are connected in the brain. Its main goal is to create a detailed wiring diagram of the brain, which we call the connectome. This diagram helps us understand not just where the connections are, but how different parts of the brain communicate. There are 3 key types of connectivity to consider. First, structural connectivity, which involves the physical connections between neurons, like axons and synapses. These are the actual wiring that links different brain regions. Next, we have functional connectivity, which looks at correlated activity between regions, so when 2 areas activate together, even if they're not directly connected physically. Lastly, effective connectivity refers to the causal influence one region has over another, how activity in one part can actually cause changes in another. Connecting these points to the broader theme of understanding the brain's complexity, connectomics helps us see the brain as a highly interconnected network. It's a crucial piece in unraveling how different areas work together to support cognition and behavior. Understanding the brain's connectivity is really fundamental to unlocking many of its mysteries. When we decode how information flows through neural networks, we gain insights into how different regions communicate and coordinate. This helps us better understand cognition, perception, and behavior, essentially the core functions that define our mental life. By mapping these connections, we can also improve our ability to diagnose and treat neurological disorders. When we know exactly how the wiring is altered in certain conditions, targeted therapies become much more feasible. And beyond medicine, understanding brain connectivity even inspires us to build smarter, brain-inspired artificial intelligence systems. These connections are not just about understanding ourselves, they also guide innovation in technology. So, in essence, connectomics is a crucial piece of the puzzle because it links the structure of the brain to its function, helping us make sense of the complexity and ultimately improving health and technology. When we look at whole brain networks, we're talking about understanding how different regions of the brain communicate on a large scale. Techniques like MRI, DTI, and fMRI are essential here. MRI gives us detailed images of brain structure, while DTI, or diffusion tensor imaging, allows us to map out the white matter tracks that connect different areas. Functional MRI, or fMRI, helps us see which parts of the brain are active during specific tasks, giving us insights into how the entire brain works together. Moving to local circuits and specific brain regions, we have more targeted techniques such as tract tracing and optogenetics. Tract tracing lets us follow the pathways between particular regions, showing us how different parts are connected. Optogenetics, on the other hand, gives us the ability to control specific neurons with light, helping us understand their roles within those circuits. When we're interested in individual neurons and their synapses, techniques like electron microscopy and patch clamp recordings come into play. Electron microscopy provides us with ultra-high-resolution images, revealing the detailed structure of neurons and synapses. Patch clamp recordings allow us to measure the electrical activity of single neurons, giving us direct insights into how they communicate. All these techniques are crucial pieces of the puzzle. They allow us to examine the brain at different levels, from entire networks down to individual cells, helping us understand the complex orchestration behind brain function. This layered approach is key to unraveling the mysteries of how our brains process information and generate behavior. Now, let's take a closer look at how the brain is organized into different networks that work together to process and transmit information. First, we have the default mode network, or DMN. This network is most active when we're at rest or engaged in introspection, like daydreaming or reflecting on ourselves. It's essentially our brain's default setting when we're not focused on the outside world. Next is the salience network, which plays a crucial role in detecting important stimuli around us. Whether it's a loud noise or a sudden change in our environment, this network helps prioritize what demands our attention. Then there's the central executive network, responsible for decision-making and working memory. Think of it as the brain's control center for planning, problem-solving, and managing tasks that require focus. Finally, we have the sensory motor network, which is involved in movement and sensation. It helps us coordinate our actions and process sensory information from our body. All these networks communicate through white matter tracks. These are bundles of axons that act like highways connecting different regions of gray matter, where the neuron cell bodies reside. Understanding how signals flow through these networks is essential, because it highlights the complex yet organized way our brain functions seamlessly to support everything from resting states to active decision-making and movement. This interconnected system truly illustrates the incredible efficiency of our brain's architecture. Now, let's look at some of the experimental tools and techniques used in brain mapping. First up is electron microscopy, which allows us to achieve incredibly high-resolution images of neurons, helping us understand their detailed structures. Then there's clarity, a revolutionary technique that makes brain tissue transparent, so we can visualize complex neural networks without slicing the tissue into thin sections. Another fascinating method is Brainbow, which labels individual neurons with different fluorescent proteins, enabling us to distinguish and trace multiple neurons simultaneously. Moving to the computational side, graph theory is a powerful framework where we model the brain as a network of nodes and edges, representing neurons and their connections. This helps us analyze the brain's connectivity patterns in a systematic way. Machine learning, then, comes into play by identifying hidden patterns in these complex networks, revealing insights that might be difficult to detect manually. Lastly, we have simulation models, which allow us to predict how signals propagate through neural networks, giving us a dynamic view of brain function. Together, these tools and techniques form a comprehensive toolkit that enables us to explore the brain's intricate wiring and understand its complex connectivity, which is central to our overall goal of mapping and deciphering brain function. Let's talk about how information signals travel in the brain through the connectome. First, we see that signals can follow different paths depending on their function. Within a single region, like the visual cortex, signals are processed locally. This is what we call local processing, where the information stays within a specific area. Then, for more complex functions, signals need to travel between different regions, such as from the visual cortex to the motor cortex. This is long-range communication, which allows different parts of the brain to work together seamlessly. Another key aspect is feedback loops, which involve recurrent connections. These loops are essential for learning and memory, helping the brain refine its responses based on experience. Now, the efficiency of this signal transmission isn't just about the paths themselves. It also depends on factors like synaptic strength, which determines how well signals are passed from one neuron to another. Myelination of axons is also crucial, as it speeds up signal transmission. And finally, the overall network topology, think hubs and modular structures, plays a big role in how effectively information flows through the brain. All of these elements come together to shape how efficiently our brain communicates, which is at the core of understanding connectomics and the intricate wiring of the brain. Let's take a closer look at disorders of brain signaling. These disorders occur when the communication pathways within the brain don't function properly. Essentially, the brain relies on intricate signaling networks to coordinate everything from movement to mood, and when these are disrupted, it can lead to a variety of neurological and psychiatric conditions. Key points to understand are that these signaling disruptions can be caused by a range of factors, including genetic mutations, neurodegenerative processes, or even environmental influences. For example, in conditions like Parkinson's disease, the dopamine signaling pathway is impaired, leading to movement difficulties. Another important aspect is that many of these disorders highlight the delicate balance required for healthy brain function. Any disturbance in neurotransmitter release, receptor activity, or signal transmission can have profound effects on behavior and cognition. Connecting this to our overall theme, understanding these signaling disorders helps us better grasp the complexity of brain function and underscores the importance of precise communication within neural networks for maintaining mental health and neurological integrity. Disruptions in signaling can have significant impacts on brain function and can lead to various neurological disorders. For example, in Parkinson's disease, we see a deficiency in dopamine, which is critical for controlling movement. This deficiency often results in elevated beta activity within motor circuits, contributing to the characteristic motor symptoms. Moving on to epilepsy, this disorder involves abnormal electrical activity in the brain. It's often characterized by excessive synchronization of neural signals, leading to spike hand wave discharges that can cause seizures. In depression, the issue is more about chemical imbalances. Specifically, there are disruptions in serotonin and other neurotransmitters, which can alter brain rhythms like alpha and theta waves. These changes are associated with mood disturbances and cognitive symptoms. Then we have schizophrenia, where disrupted gamma oscillations play a key role. These high-frequency oscillations are essential for cognitive processes like attention and perception, and their disruption can contribute to the symptoms observed in schizophrenia. Overall, these examples highlight how critical proper signaling is for healthy brain function, and how disturbances can manifest in various neurological and psychiatric conditions. Understanding these disruptions helps us grasp the importance of maintaining balanced neural activity for overall mental health. Abnormal connectivity is linked to various neurological and psychiatric conditions, and understanding these connections is key to grasping the broader picture of how the brain functions and malfunctions. For example, in autism, we see altered long-range connectivity, which can affect how different parts of the brain communicate over distances, impacting social and cognitive functions. In schizophrenia, the issue often involves disrupted functional networks, meaning the typical coordination between different brain regions is impaired, leading to the symptoms we observe. Alzheimer's disease, on the other hand, involves degeneration of hub regions. These are critical areas in the brain that serve as central nodes within the network, so when they deteriorate, the entire network's efficiency suffers. Stroke causes damage to critical pathways, disrupting the flow of information and impairing various functions depending on the affected areas. All of these examples highlight how disorders often involve connectome disruption, meaning the intricate wiring of the brain gets affected in different ways depending on the condition. Recognizing these patterns helps us see how fundamental the connectome is to healthy brain function and how its disruption underpins many neurological and psychiatric disorders. This understanding not only deepens our knowledge, but also guides us toward targeted interventions and treatments. Let's take a closer look at brain-computer interfaces, or BCIs for short. These are fascinating technologies that create a direct communication pathway between the brain and an external device. Essentially, they allow us to translate neural signals into commands that can control computers, prosthetics, or other machinery. This is especially promising for helping people with disabilities regain mobility or communicate more effectively. What makes BCIs so important in the context of technology and human augmentation is their potential to bridge the gap between biological neural activity and digital systems. They're pushing the boundaries of how we can enhance human capabilities and improve quality of life. Throughout this presentation, we've seen how these interfaces are evolving rapidly, combining advances in neuroscience, signal processing, and machine learning. The ultimate goal is to create seamless, intuitive interactions between humans and machines, making technology an extension of ourselves. BCIs are a perfect example of the convergence of biology and technology, and they're definitely a key piece in the future of human augmentation. A brain-computer interface, or BCI, is essentially a system that creates a direct communication link between the brain and an external device. What makes this really fascinating is that it bypasses traditional pathways like muscles or speech. Instead of relying on physical movement or talking, BCIs interpret neural signals directly from the brain. These signals are then translated into commands, which can be used to control a variety of devices, such as computers, prosthetic limbs, or other machinery. This technology is a key part of the broader theme we're exploring today: how brain interfaces are transforming the way we interact with technology and improve lives. So, when it comes to how BCIs actually work, the first step is capturing brain signals. These signals are then decoded to interpret what the brain is communicating. There are a few different methods for doing this, each with its own level of invasiveness and precision. The most common non-invasive method is electroencephalography, or EEG. This technique measures electrical activity from the scalp, which makes it relatively easy and safe to use. It's great for many applications because it doesn't require surgery, but it does have some limitations in resolution. Then we have electrocorticography, or this is semi-invasive, meaning electrodes are placed directly on the surface of the brain. This method provides better signal quality and spatial resolution compared to EEG, making it useful for more precise control. Finally, there are intracortical implants. These involve inserting electrodes directly into the brain tissue itself. This invasive approach offers the highest level of detail and accuracy in capturing neural signals, enabling very fine control, but it also comes with higher risks and challenges. Once these signals are captured, they don't go straight to controlling devices. They are processed using sophisticated algorithms that extract meaningful patterns, translating raw neural activity into commands that can be understood and acted upon by computers or prosthetics. So, in essence, BCIs rely on this pipeline, capturing signals, processing them, and then decoding them into usable information. This process is fundamental to how BCIs enable communication between the brain and external devices, which ties into the larger goal of restoring or augmenting human capabilities. Now, let's take a look at some of the key brain signal measurement techniques used in BCIs. First up is EEG, or electroencephalography. This method measures the electrical activity of the brain through electrodes placed on the scalp, making it relatively non-invasive and easy to set up. Then we have ECOG, or electrocorticography, which records electrical signals directly from the cortical surface, providing higher spatial resolution but requiring surgical procedures. Moving on to fMRI, or functional magnetic resonance imaging, this technique tracks blood flow changes in the brain, giving us detailed images of brain activity, though it's more costly and less portable. Next is MEG, magnetoencephalography, which detects magnetic fields generated by neural activity. It offers excellent temporal resolution, but involves complex and expensive equipment. Lastly, NIRS, or near-infrared spectroscopy, measures oxygenation levels in the brain tissue, providing a more portable and cost-effective option, though with some limitations in spatial resolution. Each of these modalities has its own trade-offs in terms of spatial resolution, temporal resolution, cost, and portability. Understanding these differences is crucial as we explore how to best harness brain signals for effective BCIs. Let's take a closer look at the core components of a typical BCI system. First, we have signal acquisition, which involves capturing brain activity through sensors placed on or in the scalp. This is the foundation because without good signal collection, the rest of the process can't work effectively. Next comes signal processing, where the raw signals are refined and prepared for interpretation. Within this step, pre-processing is crucial. It filters out noise and unwanted signals that could interfere with accurate analysis. Then, we move on to feature extraction, where we identify specific patterns or features in the brain signals that are relevant to the task at hand. This helps us focus on the meaningful information. After that, we have classification, which is about translating those patterns into actionable commands, like deciding if a person is imagining moving their hand or focusing intently. Once the system has generated a command, it goes to the device output stage, where the system executes the command, say, moving a cursor or controlling a robotic arm. Finally, feedback is provided in real time to the user, closing the loop and allowing for adjustments and learning to improve control. These components work together seamlessly to enable brain-computer interfaces to interpret neural signals and translate them into useful actions. This slide doesn't contain enough information. That's why we're moving to the next slide. The brain generates signals through two main mechanisms, neural oscillations and action potentials. These signals form the basis for how brain-computer interfaces, or BCIs, interpret neural activity. One key aspect are event-related potentials, or ERPs, which are specific brain responses triggered by particular stimuli. For example, when you see a flashing light or hear a sound, your brain produces a response that we can detect and analyze. Another important concept is motor imagery, where simply imagining moving a part of your body can activate the motor cortex. This means we can interpret these imagined movements as commands, which is especially useful in assistive technologies. Then there are steady-state visual evoked potentials, or SSVEPs, which occur when the brain responds to flickering visual stimuli at certain frequencies. These responses can be harnessed to control devices simply by focusing on different flickering options. All of these signals, ERPs, motor imagery, and SSVEPs, are captured through signal acquisition methods. Collectively, they form the foundation of how BCIs translate neural signals into actionable commands, connecting the brain's natural responses to technological interfaces. All right, so let's walk through how BCIs, or brain-computer interfaces, actually work step by step. First, we have signal acquisition, where we use tools like EEG, ECOG, or even implants to capture the brain's electrical activity. This is the foundational step, getting the raw data directly from the brain. Next comes pre-processing, which is really about cleaning up that raw signal. We filter out noise and artifacts that can distort the true brain signals. Then, we move to feature extraction. Here, we identify meaningful patterns in the data, such as specific frequency bands or spikes that are associated with certain intentions or actions. This step is crucial because it transforms raw data into recognizable features. After that, we have classification, where machine learning algorithms interpret those features to understand what the person intends to do. Finally, there's output translation, which converts these interpreted intentions into commands that can control a device, like moving a prosthetic arm or operating a computer. This entire process forms a signal processing pipeline that turns brain activity into actionable commands, connecting the user's intentions to real-world applications. It's a complex but fascinating chain that underpins the functionality of BCIs and highlights how technology can seamlessly bridge the gap between mind and machine. Let's start with pre-processing. This is a crucial step where we clean and prepare the raw signals. Filtering, such as using bandpass filters, helps us focus on the specific frequency ranges relevant to our analysis, removing unwanted noise outside those bands. Artifact removal is also vital. For example, eye blinks and muscle noise can introduce distortions, so we apply techniques to eliminate or reduce these artifacts to ensure cleaner data. Moving on to feature extraction, this is where we transform the pre-processed signals into meaningful information. In the time domain, we might look at features like amplitude, which can tell us about the strength of the signal at different moments. In the frequency domain, features like power spectral density help us understand how energy is distributed across various frequencies, crucial for distinguishing different brain states. Spatial features involve analyzing electrode location patterns, giving us insight into where in the brain certain activity is happening. Next, we have classification algorithms, which are the core of interpreting these features. Linear discriminant analysis, or LDA, is a popular method because it's simple and effective for many tasks. Support vector machines, or SVMs, are powerful classifiers that find the best boundary between different classes. Artificial neural networks, or ANNs, mimic the brain's own networks and can handle complex patterns. And with deep learning, including CNNs and RNNs, we can automatically learn hierarchical features, often resulting in higher accuracy in classification tasks. All of these steps, filtering, artifact removal, feature extraction, and classification, are interconnected. They form the backbone of signal processing in BCIs, enabling us to translate raw brain signals into meaningful commands or information. This process is fundamental to making brain-computer interfaces practical and effective. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about how we can monitor mental states like stress or fatigue without needing active control from the user. This means that systems can observe these internal states passively, which is really useful for creating more intuitive interfaces. Then, there's the ability to respond to external stimuli, such as visual flickers. These stimuli can be used to evoke specific responses or gather information about the user's state, which is especially relevant in neurofeedback applications. Finally, users can consciously control the interface through actions like motor imagery. This involves imagining movements, which can then be detected and translated into commands. All of these points highlight different ways our brain and body can interact with technology, some passively, some reactively, and some through conscious effort. The overall theme here is about expanding our interaction methods beyond traditional controls, making interfaces more natural and responsive to our mental and physical states. Let's take a look at the different ways sensors can be placed on the brain to record activity. First, we have sensors placed on the scalp, which are the most common and safest option. These are widely used in research and clinical settings because they don't require any invasive procedures. However, the trade-off is that they tend to have lower signal resolution, meaning the data they collect isn't as detailed as some other methods. An example of this is EEG, or electroencephalography, where electrodes are placed on the scalp to pick up electrical activity from the brain. Next, we have electrodes placed inside the skull, but outside the brain tissue itself. These are a bit more invasive, but offer moderate signal quality. They're used less frequently, but are valuable when more precise data is needed without the risks of penetrating the brain tissue directly. Finally, the most invasive option involves electrodes implanted directly into the brain. These provide high signal fidelity, capturing very detailed neural signals. Such methods are typically used in clinical settings, especially for patients with paralysis, where precise control or monitoring is critical. An example of this is the Utah array, which is a well-known device for high-quality brain recordings. So, as we can see, the choice of sensor placement depends on the balance between safety, signal quality, and the specific needs of the application. This progression from non-invasive to invasive methods highlights how technology adapts to different clinical and research requirements, fitting into the broader theme of developing effective brain-computer interfaces. Let's talk about some of the key challenges and limitations we face with brain-computer interfaces. First, signal noise is a big issue. EEG signals, which are commonly used, are inherently weak and can be easily disrupted by external factors or even small movements. This makes capturing reliable data quite tricky. Next, there's the issue of latency and accuracy. Since BCI systems often need to process signals in real time, achieving both quick response times and high accuracy can be quite difficult. If there's too much delay or errors, it can really impact the user experience and the effectiveness of the system. Training is another important point. Users typically need some time to learn how to effectively control BCIs. It's not always intuitive at first, so there's a learning curve involved, which can be a barrier to widespread adoption. Then we have ethical concerns. Privacy is a major issue. Brain data is incredibly personal. Consent must be clear, and there are questions about how this data might be used or misused. There's also the potential for cognitive manipulation, which raises serious moral questions. Finally, scalability remains a challenge. Making BCIs affordable and accessible to a broader population is essential, but current technology can be costly and complex to deploy at scale. All these factors highlight the hurdles we need to overcome as we continue advancing BCI technology. Let's explore some of the impactful applications of brain-computer interfaces. In medical rehabilitation, BCIs are helping restore movement in patients who are paralyzed, enabling them to regain independence. They also allow for control of robotic limbs or wheelchairs, providing greater mobility and quality of life. In communication, BCIs are making a difference for locked-in patients, such as those with ALS, by enabling them to spell out words or communicate more effectively using neural signals. Additionally, they can create smart environments where, for example, lights or appliances are controlled just by thought, offering more autonomy. When it comes to gaming and entertainment, BCIs open up exciting possibilities like mind-controlled games and immersive virtual reality experiences, making entertainment more interactive and engaging. Mental state monitoring is another key area, where BCIs can detect attention levels, workload, or emotional states. This has applications in high-stakes environments, helping to improve performance or ensure safety. Lastly, in military and aerospace contexts, BCIs enhance situational awareness and control systems, allowing for quicker decision-making and more intuitive command interfaces. Overall, these applications highlight how BCIs are transforming various fields by bridging the gap between the brain and external devices, ultimately improving lives and expanding human capabilities. Let's talk about some of the key ethical and societal implications of brain data. First, data privacy is crucial because brain data is deeply personal. It's like our thoughts and intentions, so safeguarding that information is essential to protect individual rights. Next, autonomy raises the question: who truly controls the interface? Is it the user, the technology developers, or some other entity? Ensuring that individuals maintain control over their own brain interfaces is vital. Then, we have bias and accessibility, which are about making sure that these technologies are equitable and accessible to everyone, not just a privileged few. We need to work toward solutions that prevent disparities and promote fairness. Finally, regulation is essential. There's a clear need for standards and oversight. As these technologies evolve rapidly, establishing guidelines helps ensure safety, ethics, and accountability. All of these points tie back to our overall theme. As we push forward with brain-computer interfaces, we must carefully consider their societal impact, ensuring they benefit everyone responsibly and ethically. Let's take a look at some exciting future directions in brain-computer interfaces. First, hybrid BCIs are emerging that combine multiple signal modalities. This means integrating different types of brain signals to improve accuracy and robustness, making the interfaces more reliable and versatile. Next, AI integration plays a crucial role. especially with deep learning techniques that enhance decoding capabilities, enabling us to interpret neural signals more precisely and in real time. Wireless and wearable BCIs are also advancing rapidly, focusing on creating more user-friendly designs that allow greater mobility and comfort, which is key for everyday use. Neuroenhancement is another fascinating area, aiming to boost cognitive functions beyond typical levels, opening new possibilities for mental performance. Lastly, neural implants like Neuralink are pushing towards long-term, high-fidelity interfaces that can seamlessly connect the brain to external devices, offering immense potential for both medical and enhancement purposes. All these developments are interconnected, shaping a future where BCIs become more integrated, intuitive, and impactful in our daily lives. And that brings us to the final slide. I want to thank you all for your attention and engagement throughout this presentation. If you have any questions or want to follow up, please feel free to reach out to me. My contact information is on the slide. You can email me at contact@Madany.com or Dr. Yasser, Madany@yahoo.com. You can also connect with me through my IEEE email at immersive@IEEE.org. For more information or resources, visit my website at https://immersive.com. Once again, thank you very much for your time, and I look forward to staying in touch.